So one of the things I was trying to give an analogy for is that we have in our DNA, if we take the genetic sequence and we look at this thing called a karyotype, this is all our DNA, and we have one copy from each of our parents of a chromosome. So what we can do is oftentimes in many diseases, we don't know the exact causes of what is happening. Like today, in Part's article, he was talking about interleukin-3. And that was, people are thinking that could be an early biomarker of what's going on in Alzheimer's disease. So we often don't know what are these genes and whatnot. We have over 20,000 genes. I was showing you like last week about gene expression data, about expression of different genes and trying to figure those out. So it's all a huge puzzle. So the most, sometimes when people don't know where to start, they think of the first thing, which is that the DNA makes us who we are. Essentially, your DNA, if we figured out your DNA, it makes you who you are. And epigenetics can also make the difference between, like, it's how the gene gets expressed, even with the same sequence. Only identical clones will have every single thing here the same. So this is made up of DNA. You know, if you unwind these chromosomes, they're made up of DNA that's wound tightly around these histone proteins. You know, that's what these are made up of. And when we look at it, if we're trying to analyze differences in people who have diseases and who don't have diseases, we can look at this genetic information and and we can look at the DNA and see where there are mutations that are in common. So as DNA is made up of nucleotides. Do you guys know the 4 nucleotides that we talked about for DNA? The 4 nucleotides that DNA is made up of.
They're adenine, cytosine, guanine, and thymine, right?
Yes, awesome. These are the 4 nucleotides: adenine, cytosine, guanine, and thymine. And together they make up our DNA. And they're like over 3 billion base pairs of DNA. And they make it all up. So what happens is people, if they're trying to analyze a condition, if they're trying to analyze a condition and they're trying to figure out what are the differences in people. For instance, like let's see that if this is somebody who is control, no Alzheimer's. Like let's just look at Alzheimer's disease. So this is a control, no Alzheimer's disease, and then this is somebody with Alzheimer's disease, and this is also somebody with Alzheimer's disease. So what they'll do is they'll go through the whole genome, and then they will, at all of the DNA, they'll go across all the chromosomes, and then they will look at the differences, where there are differences. So this is GCCA, GCCA, GCCA. It's all common here. So that's fine. So then they look at where are their differences. And let's say that they do this, and then they see, okay, this is an A in this person with who doesn't have Alzheimer's disease. This is a G in the person with Alzheimer's, and this is an A. Does this tell us much about Alzheimer's disease? Does it seem different? How does it seem to you? Does it seem like a good location? Or not? Like, so this person without it has an A. This person with it also has an A, and the other person has a G. Do you think that this can help us tell about Alzheimer's disease? Yes or no, guys? Do you guys think that this can tell us about Alzheimer's disease?
I would say yes.
I mean this one.
I feel like kind of, maybe. No, not that one, because like they're not the same. Like the 2 people with Alzheimer's, they're not the same.
Mm-hmm. Yeah, it may not be so great. Maybe this G could be, maybe we need more data, right, to see if maybe people have Gs as well. So maybe some people with Alzheimer's could have A and some could have G, and maybe A, maybe not having G, but we need more samples, more people. But right now, maybe we can just say, it's not so great, right? So based on this information, we might not be so keen on this, right? We just put a question mark here. But what about the next spot? Let's just say, so this is not so great because we just have 3 people, one with without it and 2 with it. So but we need lots of samples. So what about this spot? Do you guys think that this is a good spot for telling us this SNP, this mutation? Single nucleotide means one nucleotide. One of these bases is different. In a lot of people, it differs. It's different in at least 1% of the population worldwide. 1% seems small, but it's huge because many diseases are super rare and they happen in just 1% of people around the world. So 1% is still pretty high. That means it's pretty common. 1% of 8 billion people around the world is still quite a lot. So do you guys think that this is a good mutation to study for Alzheimer's disease?
I mean, there might be a chance that this is the SNPs responsible for it because, like, the female and the child has it, and the control group doesn't.
Yes, yes, thank you, Yannick, because it's A in both people with Alzheimer's disease, and it's a C in the person without it. So that could help explain, like maybe something is going on here. And then, so this seemed like a good spot. Again, this is 3 people. So you would, realistically, you would not do this for 3 people. You do this for way more people, but the same concepts can extend, right? Like you have to see, and what % of people with Alzheimer's is this without Alzheimer's is this a C and what %? So it's all about looking at like the fraction of people. So here 100% of the Alzheimer's people have, people with Alzheimer's have this A, and the other people 100% means everybody in this control does not has a C instead. So this seems like a really good spot to look at. What about this spot? How does this spot also look good to you guys? This is this spot over here.
Probably.
Oh yes, and why? Why, Ethan?
Because they're both different from the control.
Yeah, and they happen to be the same, right? So in people who have Alzheimer's, it's A, and in people without it, it's a T. So that's what this is saying. Then what we look at is this one, and this one is not good because in people with Alzheimer's disease, it can be a T or a C, and in people without it. So it doesn't give us much information about it. So we want to look for very clear patterns that can probably tell us that people with Alzheimer's disease tend to have this mutation here, like they have an A in this spot, and people without it have this version. And similarly, we want to find here that, okay, people with Alzheimer's have an A base over here instead of a T, because ultimately when we're making the messenger RNA, we're going to make different protein products. And we can have different versions of a gene that can happen because of a 1 base pair change. We were looking at those amino acid library, and if we change one of the bases, the mRNA can change. I don't want to get too confusing. mRNA can change, amino acids can change, proteins can change, or maybe a mutation can make a protein just not work at all. So the idea is in so many diseases, like neurological diseases, and you guys can check maybe for epilepsy, maybe for OCD, anxiety disorders. People are even doing these studies for orientation, like, if you're gay, if you're not, or not, or versus straight, what are the genetic changes that make people more inclined? What about for depression, anxiety? What about for post-traumatic stress disorder? For a lot of different conditions. And there are a lot of ethical concerns that are brought up as well, that what if you do genome-wide association studies and you find the spots in the DNA? So what happens is people do not know why, and some of these things are called complex diseases. Now we are trying to figure out, like, why exactly or what genes or proteins are responsible for many of these diseases. And we don't exactly know. So for people with amyotrophic lateral sclerosis, ALS, motor neuron disease, we can look at a lot of people who have the disease and a lot of people who do not have the disease and do the same type of analysis on a larger scale with thousands and thousands of people. And we can see for those who do not have Alzheimer's disease, like what mutations do they have and how are those different from, like, where in the genome, the DNA, are these different from people who have Alzheimer's disease? That's what we're trying to look at. And we can do this for anything. It's a genome-wide, it's across the genome, and it's an association study where all that's trying to do is to figure out if one DNA base being different, if that can lead to a difference, if that is different in people without Alzheimer's disease, like the C is in people without Alzheimer's disease, and this A is found in people with Alzheimer's disease. So maybe this is significant. So that's what they're trying to do. So that's exactly like if I gave you guys just everybody's genetic sequence and I asked you guys, can you guys find which, like, what are regions that are different? You guys might not think of it right now, but if I gave you enough time to think about it, I'm sure you guys would do the same thing, where you would look for spots in the DNA that are different between these two groups. And you would probably say that, hey, it looks that in many people, like in 90% of people who don't have Alzheimer's disease, they have a C here, and a majority of people, 80% of those with Alzheimer's have an A here. So this could be something huge. So you guys would probably come up with that information. If I just gave this to you and I gave you a few days to look at all of, I mean, I don't know how long, because I work with the data, but I'm sure you guys would be able to figure this out maybe. If I gave you all the data and I tell you guys, can you find differences, right? Some of you guys who know programming might write programs to just find differences between these, right? Like find the different regions between this and this. So that's what you guys would do. And then that's what genome-wide studies are basically doing. And when people find regions that are different, that's what they're plotting here. So let me show you guys. So that's what they're plotting in this. People are plotting. They what they do is, you'll see this in many different ways. This is called a Manhattan plot. But firstly, did we have any questions on that before I come here? Did we have any questions on how this SNP, these are all SNPs, single nucleotide, it's one DNA base pair being different. And the SNPs are given an ID that scientists recognize so that they can look at it. Biologists work with these. They're given something that's called like an RS number, just some SNP number if you happen to see it. All of it is saying is it's along a chromosome at a specific base pair along a chromosome. That's what a SNP is. It's a mutation. And they're just given identities. And what scientists then do is then they'll say, okay, this over here where I have the check marks, these probably are very good signs of people with Alzheimer's disease having these mutations. Of course, we would not do it for 3 people again. We would need to do this for thousands of people. But the same principles are extended just to thousands of people. So do you guys understand this so far? Why certain SNPs I said are good and why some are not? We're trying to see the ones that can be different in people with Alzheimer's versus people without Alzheimer's disease. It's like you're just trying to find what's different. So anybody have questions? If you understand this part, just put your thumbs up, please. If you guys get this part. Okay. Thank you guys. So so I'll just clear this part. But like I just want to like show you guys that why this matters, and then I'll let you guys get back into your groups because you might want to add this here. Is that people have these Manhattan plots. They're called Manhattan plots because they look like a skyline. And what happens is this is like plotting all 3 billion base pairs in the DNA, like super, like back to back to back. They use different colors like black and gray. It depends, but they're just trying to alternate. So you see this is chromosome 1, chromosome 2, 3, 4, 5. So you can tell which part is each chromosome. And this is just moving along the chromosome. So this is just moving from like the top to the bottom of the chromosome, then the top to the bottom of this chromosome, top to the bottom of this chromosome. And they're just mapping out for each of those locations that are different between people. For each of those SNPs, they are using, what this is saying is this scale. Don't worry too much about what this is saying. It's just trying to identify if this SNP, if this mutation, if it is really different between people with Alzheimer's disease or not. That's all that it's saying. So like how we found that out of those 4 SNPs I was showing you in this diagram, other diagram, 2 were significant, like they could tell us between Alzheimer's people and control people, people without Alzheimer's disease. Similarly, once we start looking at people in the thousands, so many of these genome-wide studies look at people Like this one was running in 94,000 people, 95,000 people with Alzheimer's disease and people who don't have it as well. And then they are basically looking at a group of people who have the condition, like thousands of people, and a group of people without the condition. And then looking at these differences and seeing which SNPs, which mutations are really like found in a lot of people with Alzheimer's and not found in people without it. So the higher these points are, that means that they are likelier to be found in people with Alzheimer's disease, and they can really tell between people with Alzheimer's and people without Alzheimer's disease. They may not give you the disease, they may not lead to it, but they are really high risk factors. So that's what people have found. And then they said, wow, this APO gene is like insanely high. Like if we look at, that's why people started looking in this APO gene, they identified it from here. Like that, the gene I was telling you about, the largest risk factor. I think it's super high. I think BIN is next, BIN1 is next. So then they're going to investigate it. And sometimes what they find is that here, this is chromosome 6, are a lot of immune system genes that pop up as well. And this is also an immune system gene. What happens is they then look at, and they know the exact locations of these mutations. They then go into the DNA and they look near that region. Are there any genes? Is this mutation in a gene? Is it near a gene? Like what is the function of this mutation? And in my work with Professor Wang, we're trying to figure out what do some of these SNPs that don't happen in genes, what do they do? That's what our research is trying to do. If they fall in regions that don't code for genes, how can we connect them to genes and ultimately to Alzheimer's disease phenotypes? How can we make the most out of this information? Because this is a genetic level. At the DNA level, what are differences between people with a condition and without a condition? So they often take these values, they have a threshold, and above this value, any genes that are really significant are going to be showing up here. And then you look at those genes and everything else you can ignore. So if you guys look at a genome-wide association study, I hope then you can put in the plot, maybe also take a screenshot of the image. Like, so I'll go back to this example for schizophrenia, for instance, but I'm really hoping you guys will. Yeah, I hope you guys will maybe include this, talk about how many people have the condition and that they were testing the cases and the controls. And then just talk about any of these genes that show up and just talk about like where you got this, you know, where it's from. So just kind of, and I'll let you guys go into your project groups, but sort of like for autism spectrum, maybe put in, if you can find a genome-wide study, put in the photo of the plot, or like for schizophrenia, what you can do is you can talk about, sorry about this, I just wanted to show you guys that you can just take an image of the article, put a Manhattan plot, talk about the number of people who have it, number of people who don't have it that they were testing, they'll call it cases and controls, and some key genes that they find in the article, and then you can talk about it. Sometimes some plots will be nice and they'll label it, and if you want to, you can research what these genes do. So this is just something else. I'm sure a lot of your guys' conditions could have genome-wide association studies that are done. People even do it for baldness as well, insomnia, all sorts of traits. People do genome-wide studies about people who have the condition and people who don't have it. And the one thing you do look at is that this is going along the chromosome, and we really just care about all these locations that are above this threshold. This is just the idea that like different people have like SNPs, which are single nucleotide polymorphisms. So we'll discuss like DNA as well more, but a nucleotide, we have 4, which is adenine A, guanine G, thymine, which is T, and cytosine, which is C. And we'll cover this as well more slowly. But what people are finding is that if they look across our 3 billion base pairs of DNA, and we are all like so similar, but just like the 1% makes us different, like 1% of the 3 billion base pairs, which can be a lot because we have 3 billion. So that's like millions and millions of these DNAs if we were to write our own DNA across all the chromosomes. So our DNA is packaged in chromosomes into 23 pairs of chromosomes. And what we are trying to do is we're going across the chromosomes and at specific locations. So what this is this is saying like you're going from left to right, like from the beginning to the end of chromosome 1, and beginning to end of chromosome 2, beginning to end of chromosome 3. And these are little locations. And this plot looks like it's just too much to view 3 billion locations. So they've just tried to condense these plots. I know when I was trying to generate such a plot, it took my computer hours to generate such plots because it takes a long time. But what they're trying to do is what all this is called a Manhattan plot. And it's called Manhattan because they look like skyscrapers. So you take individuals who have the disease, like Alzheimer's disease, and people who don't have it, and you try to look at differences. And the ones that, like, if almost everybody who has Alzheimer's disease has the mutations in this chromosome and in that chromosome position, then this will be super high. So these are all just mathematical ways. So the real thing is that the higher this is, like for a gene, so for the most important genes, they have bothered to label them like the SNPs. The SNPs are mutations, and they're saying that, okay, the mutations in APO gene are like extremely important, and BIN1 as well, and CR1. CR1 is a complement receptor for the immune system. So they're finding that a lot of these genes here in chromosome 6, there are a lot of the MHC for the immune system genes, and many of them are important. So people say, okay, well, we have a lot of these mini peaks. So let's have a threshold here that any of these locations that has a high enough value probability will be significant. So they're trying to see like what spots, because we have 3 billion base pairs. So it's the most, if you had to think of it, it's the most logical thing to do. If you had all these bases, how would you try to find out? Like if you could sequence out everybody with like many people with Alzheimer's, like if you have 50 people with Alzheimer's and 50 people who don't have it, if you looked in their genome and you tried to look at differences, then what these are telling you is a probability like that there's a difference between them. So the only thing to remember is that in genome studies, the higher, and these are SNPs, the SNP value is, the more significant it is. And for these significant SNPs, these locations with their mutations, people have bothered to map them to specific genes. So they were looking at this chromosome 19 here, which is where this is. This is a high peak, and they looked at its location, and they found that it falls along where APO is. It's almost like if you look at a country or the world from space, how do we draw like the shape of Wisconsin on a map, right? Like it's a huge territory, right? We needed to figure out how can we have Google Maps map everything. Similarly, we're trying to map the DNA, and it's an ongoing process because we only know about 2% of the DNA gives us these genes. So why this is important is that there are 3 different versions of this one gene. And I'll just end with this slide today, guys, just to tie in what Professor Wang was saying. So there are like just these 3 versions of this gene, E2, E3, and E4. And what happened is that this is how each gene, it gives a certain protein. And you can see like this is a 3D shape of a protein, like if you were to wrap around, and we'll discuss this more slowly. But what people are seeing is that certain versions of this gene, there are 3 different versions just based on the sequence. And that scientists have realized that there's E2, E3, E4. Even for our eye color, we have blue eyes, brown eyes, green eyes, all these different color eyes, hair color, right? These are all different versions of the similar gene, like an eye color gene. And as Alex mentioned, for height, there's so many genes responsible for that. So that's more complex. But for certain other genes, there might just be 1 or 2 of them that do a certain thing. And the different versions that we have make us all unique. So they're finding that there are 3 versions of this gene. So they're trying to investigate in Alzheimer's, this is a key risk gene. We don't know, like it's very complex. So we cannot say exactly. Some people who do not have mutations in APO, they also have Alzheimer's. So it's very like they could have other stuff going wrong with them. So we can't pinpoint one, but this is like the highest risk factor. In fact, what people are saying is that, oh gosh, If you have E2, this E2 version from one parent and E2 from another parent, what actually happens is that you are 40% less likely to have Alzheimer's disease. So it actually is super protective. It protects you against Alzheimer's from dementia. If this is the average, so typically most people tend to have E3 and E3. They tend to have this from one parent, this from another. E2 and E3 means you have this from one parent and this gene from the other parent. That's what this is meaning. We don't know if it's from mom, from dad, from mom. We don't know. So we just write it like this. So one from one parent, one from the other parent. But if you have E2 and E4, well, what do you guys think is most responsible for all? Which one seems like the worst version to have? If you had to rank them.
Double E4.
Well, yes. If you have both versions, you're 14.9 times more likely to have Alzheimer's disease. And in fact, when and Jonathan you might remember this from his presentation, that this same gene that is the highest, if you look at this, the highest risk factor, that's all you have to understand, guys. This is just the importance. The higher it is, the more important it is. And this is sort of like a very quick way, if we had to condense all of our chromosomes and DNA into like some sort of a visualization. That's what. And guys, you might think like in neurobiology, neurogenetics, understanding this is really important actually. I'm pretty surprised. Increases your risk of severe COVID-19 by twice. You're twice as likely to have severe COVID if you have this. And what this represents is this is a gene at this particular spot on chromosome 19. So if we look at amino acids as well, this is sort of what you can notice is that these are the 3 different versions of the same gene. This is just another graphic. And what do you guys notice? This is, they're pointing out the differences between the gene. So they've taken these things and they've put it into a simpler cartoon. So what do you guys notice is the difference between E2 and E3?
This SYS instead of ARG by
Right.
the yellow dot.
Right.
Mhm.
So those letters are referring to specific amino acids, which we will go over later. So this might be really confusing right now. But what this is essentially saying is when you're building a protein, like for example, if I'm building my trail mix, I could include like almonds, I could include M&Ms, I could include cashews, or I could replace the cashew with a pecan. And that would be the difference here between cysteine and arginine.
Yeah.
Is that correct here? Am I
Yes. I
biology
know, guys.
101
Oh yeah, I know, guys. This stuff might be very confusing. Don't get scared because I will come back to this again really slowly. But all I want you guys to see is that in this whole protein, the only thing I want you to take for now is, and we'll talk, is that an amino acid is just 3 DNA bases. So it's something so small, like you're talking about 3 billion bases. And this is something just so tiny that like in this one location, there are many amino acids. So look at if it's drawn to scale, this one small difference can change you from an E2 to an E3. And like a different version of the gene. And similarly, from ApoE3 to ApoE4,